So this is the simple microscope. So it is having only one lens. It is having only, come on say, one lens. It is having one, we can fix it. Why? Because there you see friends, some knobs are there, we can fix. So this is the stage and this is the gap to fix what eyepiece. So this is called as a simple microscope. And this is called as a, come on say bigger. What is this one? Compound microscope. And two microscopes are there. And this microscope is simple and this is compound microscope. And now I am explaining you brief note about compound microscope. And here we need to understand about who invented microscope. First, yes, who invented microscope? Leonardo. Leeuwenhoek. So you are saying correct answer Leeuwenhoek. So Leeuwenhoek is, why? Because I am using, and that person is not living but is dead, but he is living in our books. And he invented this microscope. And he was the first person to observe the presence of bacteria and the presence of what blood cells and the presence of human sperms using microscope. And that is the reason that person is called as father of microscopy. Who is called as father of microscopy? Leeuwenhoek. And who is called as father of biology? Sciences. You know, are you started? Okay. So here Leeuwenhoek is called as father of microscopy, but he was not first person to invent microscope. Yes.
Robert Hooke.
Robert Hooke. So Robert Hooke also not invented microscope first. And in year 1665, Robert Hooke designed the what microscope? Simple microscope, but not compound microscope. It is having some more magnification. Madam, the first compound microscope was invented by Zacharias and Johnson. Zacharias, you open your Google, there you see Zacharias and Hans Janssen, and both of them are father and son. And they are spectacle makers, specs. And they were the first person in the year 1590. Invented what? Microscope. So who first invented microscope? Come on, say bigger. Zacharias Janssen or Hans Janssen. Both of them are father and son. And they were spectacle makers. And they were the first person to use the microscope. And later Robert Hooke. And you said that Leeuwenhoek. Leeuwenhoek was the first person to observe the presence of bacteria under microscope. And also blood cells under microscope. Some minute protozoa under microscope. And human sperms under the microscope. And that is brief history. And now I am explaining you the parts of compound microscope clearly. So after my explanation, you need to come and you need to say, give a brief description about this microscope. Be ready. So women side one and also gent side one. Please to come and say after my giving what briefing. That is the demonstration about this microscope. So most of you are saying that my microscope is not working. Okay. Okay. You have microscope in your school, sir?
Yes.
Anyone? Microscope is there in your school, sir?
Yes.
And you are using microscope? No.
Sometimes.
Why? Because we are not having knowledge of how to handle the microscope. Madam, how to use the microscope? That is the problem. But microscope problem is not there. Microscope problem is not there. It is a strong one. It is made up of iron. It is not a plastic item. So this is a strong one. It is not what broken if it is fall on the ground also. Unless if you throw these lenses, they may break. But this one is not break. Okay. But the thing is, here you are observing friends of. I am telling one by one. This is eyepiece. Come on, say what is this one?
Eyepiece.
Say bigger. Eyepiece.
Eyepiece.
Eyepiece. Eye. So we are using this biconvex lens to observe specimen. And that is the reason we are using eye, and this is called as the biconvex lens. It is called as the eyepiece, eyepiece. And this is draw piece, small one, draw piece, and this is body tube. So this is a eyepiece, and this is draw tube, and this is a body tube. Listen carefully. And now I am lifting this body tube upwards. And this is the body tube, and this is draw tube, and that is eyepiece, and this is called as. So what is this one part? ये क्या बोलते हैं आप? ये आपका हाँ थ्रोट नोज अपर लैंग्वेज में ये भी हो उसका नोज है. It is called as nose piece. It is called as nose piece. You can revolve. So you can revolve this piece. So that is that it is called as a revolving nose piece. Come on, say
No.
revolving nose piece. This is called as revolving nose piece, and it is having lenses 1 and 2, and another one is empty. So here also you can fix what another lens. So these are called as objective lenses. Objective lenses. A revolving nose piece, madam. Objective lenses. Eyepiece, draw tube, body tube, revolving nose piece, and objective lenses. And this is the stage. Stage. S-T-A-G, stage. S-T-A-G, stage. And these are called as clips. These are called as stage clips. Stage clip. And this is called as handle. Handle. And this is called as a foot, base. This is called as a base. This is base, foot. And this is mirror. And this is called as lever. This is called lever. And here this is friends a diaphragm. But most of you are not knowing how to use this compound microscope. Why? Because you see now I am closing. Please come, sir. Please come. And keep your finger here slowly. What you are doing? There is a hold is not there. It is closed. It is closed. You please come, sir. It is closed. So hole is not there. Hole is not there. It is closed. So it is closed. But at the time of doing activities, you need to open it. You please come, sir. 1 second. You keep your finger. This side. Madam, you please come. This one. Keep your finger here. Take the big hole. Please come. Madam. This is important one. This is important. Keep your finger inside. So it is a hole. Now you see. Now keep your finger outside. No. Yes, that is the problem. So at the time of giving this compound microscope to you by manufacturer, and that time it is closed. You need to open it. After opening, you need to see the specimen. Do you understand, madam? That is the secrecy. That is the secret. You need to open this lever, and there you see diaphragm. You know diaphragm? So diaphragm is a layer that is separating the thoracic cavity, so chest cavity. It is a muscle, and that muscle is useful in respiration. Okay, teachers, I think you understood well about the parts of compound microscope and how to use the microscope. Already you observed a specimen under compound microscope. What is it? Hydra. So I think you clearly observe the presence of hydra, and it is having what tentacles, and it is having cylindrical body, and it belongs to phylum Coelenterata. It lives in water, in marine water, in water. It is an aquatic animal. So like that, you need to use these specimens, madam, regularly in your classroom, in your laboratory, and to make the student to understand the concepts. So different concepts are there in our textbook. So we are teaching 6th class, 7th class, 8th, 9th, 10th, 11th, like that. So each book is having what concepts are there, the units are there. Okay. In Hyderabad, we have Telangana State book, biology. Their 10th class book is having 10 units: nutrition, respiration, transportation, excretion, coordination, reproduction, coordination, life process, heredity, our environment, and also natural resource. Our environment and also concept and natural. Last one is natural resources. So like that, 10 units are there. In the same way, 6 classes are having food and components, fiber to fabric. So like that you see biodiversity. Like that you see different concepts are there. And in biodiversity, you are observing friends are different types of animals, microorganism, protozoan, and they are not visible. And there you see we are providing a box, and that is box is having what many what specimen, and use one specimen in a day and do the activity and make your children to observe the clear nature of the specimen. Okay, thank you. I think you understood well. Am I correct or not?
Yes, sir.
You understood or not?
Yes, sir.
Stop it, sir.